my name is Mia and I decided to learn about CRISPR. So I've always been interested in medicine, and recently I've been interested in the field of personalized medicine, so I decided to understand CRISPR. CRISPR is a scientific tool that can modify our DNA. But in order to understand how CRISPR works, I also needed to understand how the body works, including what's inside your cells, DNA, proteins, genes, and RNA. I also needed to understand the major scientific breakthroughs leading up to the discovery of CRISPR. So in order to present this topic, I will start with the basic information and work my way up to explaining CRISPR, how it works, and how it is revolutionizing modern medicine. So we start with the cell. For this project, I decided to focus on the ribosome, cytoplasm, and nucleus of our cell. As you can see in this image, there are many other parts within a cell that are all equally important. So I decided to focus on these three because they are the most relevant to understanding CRISPR. So ribosome plays a major role in synthesizing proteins. The cytoplasm breaks down glucose and produces amino acids, and the nucleus contains 23 chromosome pairs. Now each chromosome contains one strand of our DNA, and we have 46 total chromosomes, 23 from our mom and 23 from our dad. So next is DNA and its structure. Rosalind Franklin, James Watson, and Francis Crick all played a major role in the discovery of DNA. In 1952, Rosalind Franklin took one of the most famous pictures in all of science history. It was called Photo 51, and it was taken by an X-ray and of a strand of DNA. This is a copy of Photo 51, and as you can see, it shows an X, which proves that the structure of DNA is in a double helix or spiral shape. James Watson and Francis Crick added on to Rosalind's discovery by discovering the different nucleobases, also called nitrogenous bases, and which ones pair with each other, how adenine pairs with thymine and cytosine pairs with guanine. Each of these nucleobases, or nitrogenous bases, also pair with one sugar molecule and one phosphate molecule to create a nucleotide. And nucleotides make up our entire DNA. So genes, RNA, and proteins. Genes are small segments of our DNA, and their job is to code for proteins. And RNA is a molecule that reads instructions from DNA and delivers this information to other parts of the cell to build proteins. And proteins are found in all living organisms and are often referred to as the molecular workhorses of our cells because they run our body. There are many different types of proteins, but some examples are enzymes, which speed up our metabolism, and histones, which support chromosome structure. Protein production is a complicated process that involves two main steps: transcription and translation. In the first step, transcription, a type of RNA called messenger RNA or mRNA copies the order of nucleotides in our genes. Next, in translation, another type of RNA called transfer RNA assembles small molecules called amino acids into proteins according to the genetic code. Now, in order to understand the importance of protein and protein production, we can take a look at a type 2 diabetes instruction. So what happens when our proteins aren't programmed correctly? Well, we get sick. So in a healthy body, a type of protein called an insulin receptor's role is to transmit signals to the cell to taking glucose from the bloodstream. However, in a type 2 diabetes patient, the insulin receptors fail to properly transmit the signal, causing low insulin production, our cell not being able to process glucose, and a spike in our blood sugar. So next is DNA sequencing. It's important for scientists to know the exact order of nucleotides in our DNA because they can use this information to identify genetic patterns in malfunctioning genes. DNA sequencing is a tool that helps scientists understand how the body works. It determines the exact order of nucleotides in our DNA, which code for proteins. The order of these nucleotides is important because it specifies what the proteins will do. And if the nucleotides are in the wrong order, we get sick. The first complete strand of DNA was sequenced during the Human Genome Project. This project was funded in 1990, and for 13 years, scientists and microbiologists gathered together from around the world to study DNA. And after 13 years, they were able to accomplish their goal of sequencing just one strand of DNA, which can now be done in a matter of minutes. So the sequencing process is a complicated process. Scientists created 4 types of modified nucleotides by attaching a fluorescent dye of its own unique color to each of the 4 types. They called these nucleotides deoxynucleotides. Now, in the first step of the sequencing process, scientists split DNA into small segments so it's easier for the machine to analyze. Next, the deoxynucleotides bind to the contrasting nucleotides found in the DNA, and the whole strand of DNA is put under the machine. The machine then shines light onto this strand of DNA, and the deoxynucleotides light up different colors based on which fluorescent dye is attached to each one. Lastly, scientists map out the order of the colors to find the order of the contrasting nucleotides in our DNA. Using DNA sequencing, scientists know the exact order of nucleotides in our DNA and how our genes are constructed. So now we come back to CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats and was first discovered by Francisco Mojica in 1993. While working with viruses, he discovered that in every single virus's DNA, there's a specific order of nucleotides, which is the CRISPR, that marks the beginning of another pattern of nucleotides, which is spacer, which role is to code for proteins. He also noticed that in all similar viruses, they all had the same CRISPR and spacer sequences. Now, when we get sick, after our immune system has fought off the virus's DNA gets attached onto our DNA, so next time we get sick with this particular virus or any similar virus, we're able to produce proteins from the virus's DNA that know exactly how to fight it off. So in a way, it gives us immunity by acting like a vaccine. Jennifer Doudna and Emma Will Charpentier added on to Francisco's discovery by inventing the CRISPR-Cas system. A type of RNA called a single-guided RNA, or sgRNA, is programmed to deliver a protein called Cas9 to a specific destination in our DNA with a malfunction. Now, once they arrive at their destination, Cas9 works as molecular scissors and cuts the strand of DNA. From here, one of 2 repair systems are used. The HDR system is used if a template of the corrected or fixed sequence is provided, called CRISPR RNA or cRNA, and if it is provided, it directly implants itself into the split of the DNA. However, if CRISPR RNA is not provided, the NHEJ system is used, where the cell's repair system fixes the split in the DNA but will most likely fix it with the same malfunction as it had before. Now that scientists can map the exact order of nucleotides in our DNA and know which proteins are responsible for which bodily functions, they'll be able to take a blood sample, sequence the DNA, identify faulty DNA and which type of protein it codes for, then use the CRISPR RNA to make a template of the fixed sequence, and finally use the CRISPR-Cas system to fix the DNA. CRISPR is revolutionizing our future, and very soon it will be able to cure the deadliest diseases like cancer. I learned so much when researching about CRISPR. I learned about the cell, genes, proteins, DNA, all the different types of RNA, and the CRISPR-Cas system. I also learned how to study and research, as well as juggling this project with school, homework, and extracurriculars. Thanks for listening. Bye.